Some organisms are capable of surviving boiling water, solid ice, the vacuum of space, and even the reversal of their own biological age. These feats are the result of highly specialized, extreme biological protocols. To determine which strategy is the most effective, we have to evaluate these species using the same metrics we apply to engineered hardware. This chart tracks three specific variables: the maximum lethality an animal can withstand, the biological compromises required for that survival, and the potential return on investment for human medicine. We're going to test if these animals follow a consistent rule, whether achieving physical invulnerability requires a total pause of standard biological life. The tardigrade is smaller than a grain of sand, yet it is often cited as the most resilient animal on the planet. While humans require heavy shielding and complex life support systems to enter orbit, the tardigrade survives the vacuum of space, extreme radiation, and temperatures ranging from -272 to +150 degrees Celsius. This defense system is only activated when the tardigrade loses nearly all the water in its body. As the water disappears, the tardigrade cells begin deploying a specialized mechanism called cache proteins. As shown in this diagram, these proteins form a protective gel network inside the cell. This replaces the missing water and creates a structural scaffold that prevents the cell from collapsing under pressure or radiation. The trade-off is total immobilization. In this ton mode, the tardigrade enters suspended animation, losing all ability to move or react until the environment provides water for rehydration. Researchers are now testing these protein scaffolds to see if they can stabilize human organs for long-term storage or deep space transport without refrigeration. The tardigrade achieves physical resilience, but only by temporarily ceasing to live as an active organism. Turritopsis dohrnii is the only known animal capable of biological immortality. This is an emergency protocol triggered by starvation, physical trauma, or the completion of its natural life cycle. When the protocol begins, the adult jellyfish shrinks, retracts its tentacles, and collapses into a dormant cyst on the seafloor. Inside this cyst, a process called transdifferentiation takes over. Specialized muscle and nerve cells essentially wipe their internal programming, losing their specific biological functions. This chart maps that transition. Those neutral cells revert into stem cells, which then reorganize to build a juvenile polyp colony. The DNA remains constant throughout this transformation. One original adult becomes a colony that eventually buds off into hundreds of identical genetic clones. To survive, the jellyfish must dismantle its physical identity. It accepts structural death so its genetic information can reboot. For scientists, this mechanism provides the technical data needed to potentially trigger similar regeneration in human tissue, allowing for the repair of organs that currently cannot heal themselves. To preserve its genetic code, the jellyfish destroys its current physical form. The wood frog is the only frog found north of the Arctic Circle, where it faces inescapable freezing conditions for most of the year. For most animals, ice crystals act like microscopic blades that expand and rupture cell walls from the inside out. The wood frog avoids this destruction through an active cryopreservation sequence driven by biological antifreeze. As the external temperature drops, the frog's liver floods its bloodstream with massive concentrations of glucose and urea. This cross-section shows the glucose acting as a barrier. It fills the cell's interior, forcing expanding ice crystals to form strictly outside vital structures. The frog becomes 65% solid ice, remaining in this state for up to 8 months. During this time, the heart stops, its breathing ceases, and brain activity flatlines. The frog is clinically dead, and it relies on a massive metabolic cost to jumpstart its system in the spring. This glucose flooding mechanism is being researched to change how we store human kidneys and livers, potentially extending the time a donor organ remains viable from hours to months. The frog conquers the cold by weaponizing clinical death as a temporary, coordinated shield. Now we can compare these three survival frameworks side by side. In terms of physical resilience, the immortal jellyfish ranks in the lowest tier. While it can cheat time and aging, it cannot survive raw physical extremes like boiling or freezing. The wood frog takes the middle tier. Surviving clinical death in a body of solid ice is an incredible biological feat, but its invulnerability is limited strictly to thermal threats. The tardigrade sits at the top. Its ability to survive absolute zero, lethal radiation, and the vacuum of space provides unmatched multi-threat resilience. The effectiveness of these frameworks shifts when we look at their potential for human medicine. The ideal mechanism depends on the specific biological problem we need to solve. This decision tree maps the applications: tardigrade proteins for long-term space travel, wood frog glucose for donor organ storage, and jellyfish transdifferentiation for regrowing human muscle and nerve cells. One survives the vacuum of space, one reverses its own aging, and one reboots from solid ice. These extreme traits are highly calculated, resource-heavy strategies. Given the trade-offs involved, which framework is the most impressive? Let us know in the comments.